Hello and welcome everyone to our online Immunology Journal Club. The topic of today's talk is advances in live-attenuated vaccine development for Zika virus. The publication that I'll be reviewing in this journal club today is an open access article that is licensed under the Creative Commons BY license. The immune response to vaccination is in many ways very similar to the immune response to infection with a pathogen. And when a vaccine gets injected, this usually happens either subcutaneously or intramuscularly. The vaccine components get taken up by cells of the innate immune system, such as dendritic cells and macrophages, which then get activated and migrate to the local lymph nodes, where the vaccine's components get processed into antigens and then get presented to T cells and B cells. These cells then also become activated, and this initiates the adaptive arm of the immune response, which is characterized by the activation of cytotoxic T lymphocytes, plasma antibody-secreting plasma cells, and memory T and B cells, which form the immunological memory. The main difference in immune response to vaccination is that this immune response is initiated very effectively, and it usually does not cause any symptoms in the vaccinated individual. However, different types of vaccines can be better or worse at eliciting the different components of the adaptive immune response, and we will talk about this a little bit more in the next slide. There are three main types of vaccines for viruses. The inactivated vaccines are based on viruses that have been killed either by heat, chemicals, or UV irradiation. And this renders the viruses, of course, non-pathogenic and also replication incompetent, but they can still induce an immune response that protects from future infection with live virus. The live-attenuated vaccines are based on viruses that have been compromised, and this renders them non-pathogenic, but they can still replicate and induce a protective immune response. These live-attenuated viral vaccines can be either derived from naturally occurring, closely related viruses that are non-pathogenic. They can be derived by serial passaging in culture, or they can be derived by genetically attenuating the pathogenic virus. The third main type of vaccine for viruses are the subunit vaccines, and these can be further subdivided into protein and nucleic acid-based subunit vaccines. The recombinant protein-based subunit vaccines are viral proteins that are expressed either in cell lines or by yeast, and they're often given in combination with an adjuvant. The non-pathogenic vector virus-based vaccines are based on a non-pathogenic vector in combination with the pathogenic viral DNA. And one recent example is the Ebola virus vaccine, which is based on the vesicular stomatitis virus backbone. There are the plasmid-based subunit vaccines, which is a plasmid with a gene from the pathogenic virus inserted, and these are currently only available as veterinary vaccines. And there are also several mRNA-based subunit vaccines that are currently still in development, with one very important one being the mRNA vaccine from Moderna that is currently being tested for SARS-CoV-2 vaccination. Importantly, these different types of vaccines are somewhat different in their ability to induce the cell-mediated and antibody-mediated immune responses. Typically, the inactivated and subunit vaccines are not very good at eliciting the cell-mediated immune response, but they are still very potent at eliciting the antibody-mediated immune response. And live-attenuated vaccines are very efficient at eliciting both cell and antibody-mediated immune responses. Live-attenuated vaccines that are the topic of this journal club today have been around for a very long time, but they are still often used as the method of choice for vaccine development because they have several very important advantages. They are very inexpensive to produce, and they can induce a quick and strong immunity. There is a fairly low risk of adverse effects, and as I just mentioned, they can activate all phases of the immune system, the cell as well as the antibody-mediated immune responses. They are also very good at eliciting a long-lived and targeted immunity. There are some disadvantages of live-attenuated vaccines. The production process can be fairly lengthy, and there is also a small chance that the attenuated virus can undergo phenotypic or genotypic reversion, which would render it pathogenic. As it is a live vaccine, there are some issues with vaccine stability as there is some temperature sensitivity, and they can be potentially inappropriate for immunocompromised individuals, as these individuals cannot necessarily mount a protective immune response with the same efficiency. And also importantly, live-attenuated vaccines are usually not given during pregnancy, and this is because of the altered immune status during pregnancy, which I will go into a little bit more detail in this next slide. Pregnancy is a special situation for the mother's immune system, as the fetus expresses paternal genes that can potentially induce a immune response in the mother that would reject the fetus. So the immune functions in the mother are somewhat restrained to allow for fetal tolerance. And this is characterized by, for example, the reduced cytotoxicity of natural killer cells. And importantly, there is also a change in the balance of T helper cell responses. In pregnancy, there is an enhancement of Th2 responses and a suppression of Th1 responses. And this leads to an enhancement of antibody-mediated immunity and a suppression of cell-mediated immunity. Also, there is a progressive proliferation of T regulatory cells, which further suppresses immune responses in pregnant mothers. And this altered immune status makes pregnant mothers more susceptible to infection with some pathogens, and notably viruses, because successful viral clearance is dependent on Th1 responses. And this brings me to the paper that I'll be reviewing today, where the authors investigate the safety and efficacy of a live-attenuated vaccine during pregnancy. And this article was published in Nature Communications in 2019 by Chao Shan et al. And the title is Maternal Vaccination and Protective Immunity Against Zika Virus Vertical Transmission. Zika virus is a mosquito-borne virus, and it causes congenital Zika syndrome in pregnant women, and it can also cause Guillain-Barré syndrome in adults. There were several major epidemics in 2015, which prompted rapid vaccine development, and the development of an effective vaccine for Zika virus seems feasible because several effective vaccines for 4 other flaviviruses have been developed and licensed, and these are all based on live-attenuated or inactivated vaccine technology. There have been several vaccine platforms developed for Zika virus, and some have now entered clinical phase 1 and 2 trials. And these are the DNA subunit vaccines, VRC5288 and 5283, GLS 5700 and the formalin inactivated virus ZPIV. However, it still remains to be seen how long-lived the protective immunity induced by these inactivated and DNA subunit-based vaccines is. The group that published this paper, that is led by Pei-Hong Shi, studies flaviviruses that cause significant human disease. And they have previously developed a live-attenuated vaccine candidate for Zika virus with a 10 nucleotide deletion within the 3' UTR of the Zika virus genome. And they call it 3' UTR delta 10 LAV, or in short, delta 10. And they have previously shown that immunization with delta 10 protected mice and non-human primates from Zika virus infection and in utero transmission. And the vaccine also could prevent male reproductive tract infection in animal models. The maternal vaccination during pregnancy for Zika virus has not been directly evaluated. So in this current paper, this group sought to study the safety and efficacy of the delta 10 vaccine in the A129 pregnancy mouse model. And these mice are deficient for the interferon alpha beta receptor, and this makes these mice susceptible for Zika virus infection, and they show symptoms of Zika virus disease, which is not observed in wild-type animals. In the first experiment, the authors wanted to investigate whether the delta 10 vaccine is safe for maternal vaccination. And for this purpose, they mated female mice with male mice, and on embryonic day 0.5, they infected the mice with wild-type Zika virus or the delta 10 vaccine. And then on E18.5, they harvested maternal and fetal tissues and looked at the viral load, the fetal body weight, and the maternal and fetal neutralizing antibody titers. They observed, as you can see in the graph on the left, that in the group that was infected with wild-type Zika virus, 54% of fetuses were resorbed, whereas in the delta 10 vaccinated group, only 10% of fetuses were resorbed, which was similar to the mock-treated group. When looking at fetal body weight at E18.5, they saw that in the wild-type Zika virus infected group, the fetal body weight was reduced, whereas in the delta 10 infected group, the fetal body weights were similar to the mock-treated group. So they concluded that the delta 10 infection did not affect fetal resorption or body weight. They also looked at the presence of infectious virus in maternal spleen, brain, placenta, or fetal head, and they did not detect any infectious virus, neither in the wild type nor in the delta 10 infected group by their viral plague assay. When they used the more sensitive qPCR assay to look for viral RNA, they saw that viral RNA was present in every placenta of the wild-type infected virus mice, as you can see in the graph on the left, whereas none of the delta 10 vaccinated mice had viral RNA in their placenta. And in neither of the groups, they were able to detect any viral RNA in the fetal head. When looking at the neutralizing antibody titers, they saw that there were robust neutralizing antibody titers present in both dams and fetuses from both wild-type and the delta 10 groups. And in a separate experiment, they looked at whether maternal vaccination had any adverse effects on the pup's body weight, length, or behavior after birth, and they could not see any adverse effects. Next, they wanted to see whether a single dose of delta 10 vaccination is effective in protecting mothers from viral challenge. And for this experiment, they immunized pregnant mice at E0.5 with 10 to the 3 focus-forming units of the delta 10 vaccine. Then on E10.5, they bled the mothers for neutralizing antibody titers, and on the same day challenged them with the Puerto Rican wild-type Zika virus strain. 2 days later, they looked for viremia in the mothers, and on day 18.5, they harvested the maternal and fetal tissues to look for viral load and neutralizing antibody titers. As you can see in the graph on the left, when looking at maternal neutralizing antibody titers at E10.5, they detected no neutralizing antibodies in the placebo group, whereas there was a robust induction of neutralizing antibody titers in the Delta 10 vaccinated group. When looking at maternal viremia, they saw that the Delta 10 vaccination was able to prevent maternal viremia completely upon virus challenge. And when they were looking at maternal tissues at E18.5, they saw that there was no infectious virus present in any of the maternal organs in the Delta 10 vaccinated group, with the representative graph for the placenta shown on the right. They also looked at the presence of infectious virus in the fetal head, and again, they did not detect any infectious virus in the Delta 10 vaccinated group. When looking at the fetal body weight, they saw that the Delta 10 vaccination could prevent the reduction in fetal body weight that was observed in the placebo group. When looking at the neutralizing antibody titers, they saw that Zika virus challenge did not increase the maternal neutralizing antibody titers when compared to the just placebo-treated group. So they concluded that in pregnant mice, sterilizing immunity was already achieved after Delta 10 vaccination. And they also saw high maternal neutralizing antibody titers in the fetuses from vaccinated dams, as you can see in the graph on the right. And they concluded that neutralizing antibody titers get transferred from mother to fetus. They also did a separate experiment where they did later vaccination, and they saw a similar protective neutralizing response. Next, they wanted to see whether the maternal vaccination had an effect on offspring protection. And for this experiment, they again immunized pregnant mice at E0.5 with Delta 10, then bled these pregnant mothers at E10.5 for neutralizing antibody titers, and let the pregnancies come to term and let the mothers deliver the pups, and then bled the mothers for neutralizing antibody titers on day 21. They then divided the newborn pups into 3 groups. Group 1 was just followed for 1 day, and then the fetal blood and mother's milk was assessed for neutralizing antibody titers. The group 2 was challenged on day 1 with a low dose of wild-type Zika virus and then followed for 21 days. And the third group was assessed for neutralizing antibody titers weekly until day 70, when the pups were challenged with a high dose of wild-type Zika virus and then assessed for viremia 2 days later. Looking at the first group, where they just looked at the neutralizing antibody titers in 1-day-old pups' blood and maternal milk, they saw that 1-day-old pups born to Delta 10 vaccinated mice had a high blood neutralizing antibody titer in both blood and maternal milk. In the second group, where the 1-day-old pups were challenged with a low dose of Zika virus, they saw that in the pups that were born to the Delta 10 vaccinated mothers, all of the pups survived the viral challenge until day 21, whereas in the placebo group, all pups succumbed to virus infection by day 6 post-infection. When they followed the kinetics of neutralizing antibody titers over time in the pups, they saw that the neutralizing antibody titers in the offspring gradually waned from day 21 to day 70. And when they then challenged these 70-day-old pups with a high dose of Zika virus, they saw that this resulted in viremia in 68% of animals, but there was a much lower viral load in these pups born to Delta 10 vaccinated mothers when compared to the placebo group. So there was still at least partial protection by the antibodies that were transferred from the mothers to the pups. Next, they wanted to see what the minimum necessary neutralizing antibody titer was to protect the mothers from viral challenge. And for this purpose, they did a passive transfer experiment where they took immune sera from mice that had been previously immunized with Delta 10 and transferred these immune sera to pregnant mice at E9.5. They then led the pregnant mothers 1 day later to assess their neutralizing antibody titers and then challenged them on the same day with wild-type Zika virus. They assessed viremia 2 days later, and then on E18.5, they again harvested the maternal and fetal organs. So they subdivided the mothers based on their neutralizing antibody titers as determined on E10.5 into 5 groups with differing neutralizing antibody titers, as you can see in the x-axis of these two graphs. And then they looked what the minimum neutralizing antibody titer necessary was to protect the mothers from viral load in these different tissues. They saw that a neutralizing antibody titer of 1 in 100 was necessary to protect the mice from viremia, and a higher neutralizing antibody titer of 1 in 460 was necessary to protect the mothers from viral load in the brain, spleen, and placenta. And also that same minimum antibody titer of 1 in 460 was necessary to prevent fetal weight loss. Interestingly, as observed here in the graph for the maternal spleen, they saw that low levels of transferred neutralizing antibodies led to higher viral loads in the spleen when compared to the placebo group. And they speculated that this might be due to antibody-mediated enhancement. They next looked specifically at the T-cell response to Delta 10 vaccination. And for this experiment, they compared a pregnant group to a non-pregnant group, which they treated in a similar way. Again, they immunized the mice with Delta 10, and then at E18.5, in the case of the pregnant mice, they harvested the spleens and did their T-cell analysis, and the non-pregnant group was vaccinated and analyzed in a similar manner. So they removed the spleens and then isolated the splenocytes, and in culture stimulated the splenocytes with either live Zika virus or a viral E-peptide. And then they looked for intracellular cytokine responses and also secreted cytokines. As you can see in the two graphs for CD4 T cells on the left and CD8 T cells on the right, they saw that Delta 10 vaccination led to robust Zika virus-specific interferon gamma and interferon gamma TNF-alpha double positive CD4 and CD8 T cell responses. Interestingly, they saw that in the non-pregnant vaccinated group, they had stronger induction of interferon gamma in both the CD4 T cells and CD8 T cells. And they also had a significantly stronger induction of both interferon gamma and TNF-alpha in the non-pregnant vaccinated group when compared to the pregnant vaccinated group. They also observed a similar pattern when looking at the secreted cytokines in pregnant versus non-pregnant mice, where they saw that non-pregnant mice secreted higher levels of IL-2 and interferon gamma than pregnant mice. So they concluded that the T-cell response to delta-10 vaccination is weaker during pregnancy. And this is consistent with what we discussed in the beginning, that in pregnancy, the cell-mediated immunity is suppressed. To summarize, the key findings in this paper are that maternal vaccination with delta-10 is safe and did not yield any adverse effects on pregnancy, fetal development, or offspring behavior. The maternal vaccination with delta-10 prevented maternal infection and also in utero transmission of Zika virus during pregnancy. The neutralizing antibodies were transferred from mothers to pups and also protected the pups from Zika virus infection after birth. And the passive transfer of antibodies from immunized mice could also confer protection from Zika virus infection. And lastly, the T-cell responses to delta-10 vaccinations were weaker in pregnant versus non-pregnant mice. There are several open questions that still remain to be answered. For example, do the immune responses to delta-10 observed in mice directly translate to humans? And this would be important to examine as the placental physiology and also the antibody transfer are fundamentally different in mice versus humans. Further, it needs to be seen whether the delta-10 vaccine has any adverse effects on human embryos at different times of gestation. And it needs to be seen whether the other non-replicative vaccine platforms are potentially more appropriate for pregnant women. And even though these other vaccine platforms may require multiple rounds of immunization, the human gestational period would allow for that. We don't know yet how durable the sterilizing immunity conferred by delta-10 vaccination is. And also, it will be important to investigate whether delta-10 vaccination has any adverse effects on subsequent infection with other flaviviruses, as some cross-reactive immunity has been observed between dengue virus and Zika virus. The delta-10 vaccine candidate is now being advanced to clinical trials, and it will have to be seen in the future whether we maybe need multiple complementary vaccine approaches to cover our various target groups, where the inactivated or subunit vaccines might be more suitable for immunocompromised individuals and pregnant women and aged individuals, and the live-attenuated vaccines will still be very valuable for non-pregnant healthy individuals and children living in or traveling to Zika virus endemic countries, because this type of vaccine is very cost efficient, and also the single dose administration will be very practical. This concludes my talk for today.